So we're gonna welcome Heather Callahan. She's gonna be presenting virtually.
Perfect. So, hi, everybody. I'm sorry I can't be there in person. Although I lived in San Diego for, I want to say 8 years, I currently live in Ohio. So I'm on the East Coast time. It's 7:20 or something at night for me, 7:15. Um, and I'm doing this by Zoom. So I'm sorry I can't be there. I'm the CEO of Allander Biotechnologies. We are developing a first-in-class topical protein therapeutic for the treatment of oral mucositis as well as multiple skin indications. And our interest is, and you'll see this as we go through the deck. I'm going to talk to you about our company and what we do. But overall, our interest is building our advisory team around in prior industry professionals who've gone all the way through raising money, successful clinical trials and things like that in various areas. And so I'm hoping to network and get to know people. Let's see. Okay, so I can't see anybody there, so it makes it very hard. It's very difficult to tell where you're understanding or not understanding anything. So hopefully I will make it clear enough and not bore you at the same time. We all know how exciting it's been over the last few years with all of the new cancer therapies that are on the market and are helping patients, improving their, their survival rate for that matter. We're focused on being the add-on around that. And so not just improving their survival rate, but also improving their quality of life. One of the major side effects of a lot of these cancer therapies is horrible mouth, oral sores. So, canker sores in your mouth, but just truly horrific. And they're severe enough that patients end up unable to eat or drink. They end up hospitalized. And it can impact the ability of the patients and the clinicians to actually treat the cancer, to give the patients optimal cancer care. And this isn't rare. It happens in over 400,000 patients annually. 90% of head and neck cancer patients, 50% of breast cancer patients. So it's frequent and it's a big deal for those patients and for their treatment. It's also a really large market. It's a $1.5 billion market in 2024. And despite the size of the market and the impact on patients and the numbers of patients that it impacts, there's no therapeutic for 96% of patients. The last therapeutic that was approved was over 20 years ago, Kepivance, and it was only approved for use in 4% of patients because it carried a risk of actually causing cancer, right? Not what you want to give to cancer patients. And so this graph on the right, this pale pink bar here shows the size of the market that Kepivance has because it's the only therapeutic. The rest of these bars are generally palliative care, so they're barriers to radiation. There can be analgesics, the antimicrobials, and things like that. And so our belief is, in talking to clinicians and patients, they need therapies that accelerate healing, that will allow optimal cancer care and that enhance their quality of life. And that's what our topical AllaThera does. So and I'm going to give you a little bit of science, but not too much, just to help with figuring out who might be good partners with us. This is based on a protein. It's called SMAD7. We've identified the functional portion of that protein and fused it with a cell-penetrating peptide. That allows us to have a drug that is a topical drug. So in this case, the patients, it's given to the patients as a rinse, as kind of a mouthwash type approach, and they swirl it around their mouth for a minute. And that allows the protein to enter cells on contact and act right where the patients need it. So it doesn't go systemic. And we've done our GLP systemic tox and showed that we don't have any systemic side effects from this drug as well. We know a lot about the mechanism of action. We know it acts through multiple pathways. Because it is an intracellular protein, it enters cells on contact. It then traffics between the nucleus and the cytoplasm. It has activities in both places, with the result being that it decreases. So all the things you want to prevent and to treat wounds, the oral ulcers, it decreases inflammation, oxidative stress, fibrotic response, while at the same time, it's increasing epithelial cell proliferation and migration. So if there is damage and there is a wound, it will heal it. It increases DNA damage repair and decreases netosis. Netosis is another cause of damage in these kind of inflammatory situations. And so again, just to be conscious of time and not having any idea, I couldn't really hear your introduction, so I don't have a sense of your backgrounds, but I'm going to assume a lot of you aren't scientists. So I'll just say what we're really looking at here is that we've done a mouse model of cancer where we've shown that if anything, our topical treatment for oral mucositis potentiates with the cancer therapy to kill more cancer cells. So that's a good thing. And at the same time, it actually prevents killing of cells in healthy tissues. So it's acting exactly as you would like the drug to act when you're using it for cancer patients. And that was done in mice, a mouse model. We've also looked at it in a dog model. And what we found here is that if you provide it even before the ulcers have formed, it actually prevents progression of any ulcer. And that's in contrast to if you're just giving vehicle, ulcers will form in this model. Again, something that patients would be, if this also replicates in patients, something they would be very grateful for and would help them with their cancer therapy. We've looked at all the PD markers. Again, it's mimicking what you want it to do, what we've seen in humans and what you want to have happen in humans. We're at the point where we are manufacturing our clinical batch currently. We'll be filing our IND in Q3, and then we would be starting our first clinical trial at the end of this year, beginning of next year. It's a dose escalation clinical trial, and it would be in head and neck cancer patients who are suffering from oral mucositis. So how do we look at the market? How do we set ourselves apart? And one of the big things that I already talked about is that we're acting through multiple different pathways, both to prevent damage to the oral mucosa, but also to enhance healing if any damage does occur. And we've compared that to Capavance, which is the one drug that's on the market. And you see that it really only acts through one pathway. And then some of the other ones that have been in clinical trial through phase 2 or even phase 3 in the case of this one, avasopasm. And in each of these cases, they may have one or two pathways that they're acting through, but they're not acting across all the different pathways that we do. And we see this as a big differentiator, but in addition, it's one of the reasons that we think we'll succeed where they have not. As I mentioned, we also have a lot of animal data, efficacy data for skin indications, and we're ready to start clinical trials there as well as soon as we do a GLP cutaneous tox study. I should note that we have nice data in a rare disease model that would qualify for orphan drug authorization from the FDA. So that's likely to be one of the first indications that we go into. We do have a patent portfolio. It covers the composition of matter, formulations, as well as methods of use. And then this is why I'm here. We have a very small team. It's me and then the academic inventors and founders, Dr. Xiaojing Wang. She and I were together at the University of Colorado, which is where I met my Feng and Leah. And then she's moved to UC Davis, where she's the CSO of the Comprehensive Cancer Center there. She and her team and we're that's all of Allander. And so we know that we need to bring on people who have done this before. So up to now, what we've focused on is we have a good advisory team that we've worked with really closely, and they've made all the difference for us. But we know that the next step is actually bringing on industry experienced people who've already gone through this. And so just to wrap up, I know I was supposed to take 10 minutes, and I think I'm just about there. Where we are right now is we're looking to start our phase 1A clinical trial later this year. We have the funding for that. And then we're in the middle of a raise trying to find funding to do eventually our phase 1B2 expansion, as well as to do the GLP cutaneous tox. And I'll just end by saying we've done all of this primarily with non-dilutive funding. We've had 11 million in grant funding, and then with some key equity funding of just over 5 million. That now we need to raise a lot more than that. So, and I'll stop there. Love to take questions and love to connect with people.
Awesome. Any questions for Heather in the room? Go for it.
Can you hear me on the?
You're incredibly faint.
Okay, can you hear me now?
That's better, thanks.
Okay, well, I'm not a scientist at all, have literally zero knowledge, but just curious, like what's the difference with the cancer-caused sores versus the normal ones? And if there wasn't any drug for the last 20 years, how do you think the patients have been managing? Is it just over-the-counter drugs? And if that's the case, then how would you compete there? And maybe it's totally irrelevant question, and then you can just shut me down.
No, they're great questions. Thanks. Trying to remember them in order. I remember the second one, so we can start there. Actually, I remember the first one. So we anticipate that whether the oral sores are caused by radiation or other cancer chemotherapies or immunotherapies or completely different causes, that all of them would be treated. They're basically the same. All of them would be treated by our drug. The only thing that ours isn't going to do is if it's virally mediated, ours is not going to treat the virus that's caused it or or a bacteria or whatever. But if otherwise any kind of sore that you randomly get, there isn't a difference, and it would, from what our data, it would heal. Your other question was, so right now what patients do and what physicians do is they write a prescription for what's basically a mouthwash that doesn't have a therapeutic in it, but it helps the patient's mouths feel a little bit better. It's the best that they have to offer, and so that's what they offer. There's also on that table, there are barriers that people try, and ice chips and lasers, and they try anything that they can. We would anticipate that people will still use those, and that's fine. We think they'll be complementary to what we do. And so even in our clinical trial, we're not trying to prevent people from using these other things in combination. It's just the reality is that they don't stop it, and they don't actively heal anything. They're palliative. They help people feel a little bit better. So yeah, does that help?
Yes, thank you.
I have a question about the mechanism of action you explained. So this is a peptide-based therapy then, that the peptide is specific to tissue repair or healing?
Yes, so it's a protein, SMAD7. We've taken the functional portion of the SMAD7 protein, fused it with a cell-penetrating peptide. SMAD7 has, so Dr. Jiajing Wang has studied SMAD7 over many, many years. And so she knows, and it's published, a lot of the mechanisms of action of SMAD7. And we've documented that that this protein together with this cell penetrating peptide that just lets it get into the cell again recapitulates those activities of the SMAD7. So we know that in the cytoplasm, it binds SMADs, which is how it inhibits TGF-beta pathway. And that's one of the pathways that blocking it reduces inflammation. It also has roles in the nucleus, causing either binding with various factors that then change transcription rates. And each of these has been mapped out to understand what impact that has on the various pathways. So it's not a peptide, but it is a protein. And so it's doing what it normally does, except we've given extra of it. And so it does what it's supposed to do in that circumstance.
Thank you.
Thanks.
Can you hear me?
Just barely.
Okay. You said 400,000 people a year get oral mucositis. Is that right?
That's the cancer patient market of oral mucositis, yes.
How many of those people, 400,000, are going to the hospital because they have the oral mucositis that's not treated? I'm trying to the depth of the need.
Sorry, I missed the last part of that.
I'm just trying to understand the amount of need. So I'm just wondering, out of the 400,000, how many of those are going to the hospital because they can't eat?
I don't think that we have, I don't have that data. I'm not sure that that's collected anyplace specifically. The goal from the clinicians and the patients is not to ever let it get that severe. And so they will change the planned treatment profile in order to prevent that from happening. What they typically do is they'll, by default, just put in, they often will just put in the feeding tube just in case. But I don't have the specific numbers on that, on how many that is. I guess the other part of that is we wouldn't just be treating the patients who would be going, you treat sooner than that so that they never get to that point. And even the ones who wouldn't ever get that serious would also get benefit from either preventing it from going further or from accelerating the healing of the oral sore.
Okay, thank you. Can I just do one more follow-up?
You're really, really soft.
Does this work? I don't know. Okay, do you have comparative data in your animal models with the current therapy that's on the market?
Yes. Yes, we do. And so we have that. It's actually published in one of our first papers a few years ago, showing that ours works both in prevention as well as in treatment. Ours works topically, theirs doesn't, and also showing that we do not cause cancer, and if anything, we potentiate the cancer killing of the chemotherapeutics or the radiation treatment.
Thank you.
Hello.
Hi.
And I'm curious, and if you struggle with that, since it's going into the mouth with saliva, and it's a pretty intense environment for that protein.
I think I missed this first part of the question. Can you repeat your question? It's really hard to hear sometimes with the microphones.
All right, can you hear me now?
Mm-hmm.
Okay. My question was around protein degradation and how you're able to overcome that since the protein needs to remain intact in saliva, and how you work through the correct dosage as well. Just more curious.
Yeah, no, great questions. So one of the main reasons that this hasn't been a problem is that as soon as the protein is in contact with cells, so the mucosa, it will enter it'll cross the cell membrane and enter the cells, which is where it needs to be. So it doesn't spend a lot of time in just floating around in the saliva, because you're right. I'm not 100% sure if we've specifically looked at what happens to it if it does spend more time in the saliva. But we've done it in the oral cavity of mice and dogs at least, and so that is how we've identified the dosing that's needed. So we know what we used for each of those dosages and can appropriately scale it for humans. But that is one of the reasons for the phase 1A clinical trial, is to try some of the doses and make sure that we have accurately identified the correct dose.
Yeah, thank you.
And we have one question from Josh, and he's virtually. Josh, go ahead.
Okay, thanks, Heather. Wonderful presentation. Just a quick question, just from your transition into phase 1, preparing to raise for phase 2 studies, from what I recall in your meeting. What are your thoughts around how you're going to strategize that fundraising process? And do you feel as though, because from a de-risking point, some of us would want to wait to see what the phase 1 data would look like before being interested in fundraising for phase 2. I guess, what is your thought process behind getting from phase 1 to phase 2, assuming that the data looks the way you guys are expecting, given the mechanism that you've described?
Right. Yeah, I think that's a great question. So really, it had to do with the fact that we were able to get non-dilutive funding to support the phase 1A.
Mm-hmm.
And so we didn't need to raise specifically for the phase 1A. However, we do need to raise additional money after that. And the way we had been looking at it was that by being able to do our GMP manufacturing, to have completed everything, have our IND filed, and be able to start our phase 1A, have our clinical trial lined up at UC Davis, have the site and all of the IRB approved and all the rest of that, that those are really the things that the investors are looking at in order to come in for the next investment. Waiting until, so our options would be, even if the investment didn't happen until it could be tranched, or didn't happen until the end of the phase 1A, we wouldn't want to start raising then. The phase 1A should be completed in a year. And it takes a little while to do a raise. So then we'd be sitting not able to do anything until we raise money, if we waited to start the raise until the phase 1A. And I think, and the other reason is that there are a lot of other things that it can also support, such as the GLP cutaneous tox, which we could be doing while the phase 1A is running. And that would then get us prepared to also be able to start, when we had funding for it, start a clinical trial in a skin indication as well. There's a lot of interest from companies in that area, interest in partnering with us once we have that filed. So one of the things that we've been thinking about is, to your point, does it make sense to do a smaller raise, another smaller raise right now, rather than the larger raise? And so the smaller raise could be specifically to do the phase 1B, which won't take that much more money, and it could fund the GLP cutaneous tox, and, let us do another batch of manufacturing, things like that. So some of those earlier steps. So that's another approach. And we're really interested in understanding, would that be more interesting to investors right now to do a smaller raise? Because as you said, we're on the cusp of having the data for in patients, but we don't quite have it yet.
Right. Okay. Yeah. Thank you. Thank you.
Thanks.
Awesome. Thank you very much, Heather, for presenting today.
Thanks, everybody.
So now we